So we are going to start with online classes for paper MBH 402 Industrial Microbiology. There are nearly 6 units. We are going to start with unit 1, which deals with the introduction on industrial microbiology, the scope and how microorganisms can be improved and how they can be preserved, they can be contained in an industrial environment. So industrial microbiology is a subject wherein we study about the large scale and profit motivated production of microorganisms or their products for direct use or as precursor for other end products. From ancient time we have been using microorganisms to produce various products like alcohol, vinegar, bread and other various fermented products in every other countries. But with awareness about microbes and their ability to produce various metabolites sparked the development of the field of industrial microbiology. So central to this industrial microbiology is the term fermentation. Fermentation, as we all know, biochemically we can define this term as an anaerobic process wherein the sugar is converted to end products like ethanol, acetic acid, butyric acid or propanoic acid is called as fermentation. That is, the process doesn't enter into the TCA cycle or it doesn't go into the oxidative phosphorylation. That is what we call it as fermentation. But for an industrial microbiologist, fermentation is a very broad term which means any product produced by mass cultivation of microorganism is called as fermentation. So it is nothing but a process. For an industrial microbiologist, any process for the production of any product by mass cultivation of any microorganism is defined as fermentation. So it's a very broad term, and fermentation technology as such implies the role of microorganism in an industry. How at an industrial level we utilize microorganism for production. That is what we deal in fermentation technology. If you look at the chronological development of industrial microbiology, there are 5 eras. We call it as pre-1900 era, wherein large scale production of, not large scale as such, but production of alcohol and vinegar used to happen before 1900, mainly without the knowledge of microorganism. Usually it was done in wooden barrels and it was called as batch fermentation, and it was a small scale production, usually in wooden barrels. But from 1900 to 1940 it was an era of World War I, during which there was a requirement for explosives. So microorganisms was utilized for production of glycerol, citric acid, acetone, butanol, which was raw material for explosives. And during this period it was done in steel vessels. Usually batch fermentation, fed batch fermentation was followed during this era. From 1940 to 1960s, it was a period of World War II wherein people has to be treated with wonder drug called penicillin, and for this large scale fermentation process was followed. This was a period when aseptic process was followed and mechanically aerated vessels were used. Continuous fermentation process was introduced during this time. Apart from antibiotics, namely penicillin and streptomycin, amino acid production and nucleotide production was happening in large scale. 1960 to 1980 is a period for, we can call it as a period of world famine, wherein there was a shortage of food. So the industry mostly targeted on single cell protein production. During this period, continuous fermentation process was utilized. Automated control of fermentation was the highlight of this period. Automated control means the fermentation vessel was connected to a system, and the system monitored the entire fermentation process. So manual work was reduced and the process became automated. After 1980, that is 1980 to till date, it is an era of genetic engineering wherein, for example, we are making E. coli produce insulin, so we call it as heterologous products. Here we are using we are using both or all types of fermentation process in use: batch fermentation, fed batch, continuous fermentation. Fermenters are finely developed and they are being used. Now, as of now, if you look at the scope for an industrial microbiologist, we have scope in four areas, main areas. That is one wherein microorganism itself is a product of the fermentation. Second, microorganisms producing various metabolites. I think microorganisms producing enzymes can be separately categorized as 2, and microbial metabolites as 3, and micro recombinant products is one more area of industrial microbiology which is gaining more importance nowadays. And fourth is microorganisms are involved in modifying a compound that is added in the fermentation. We call it as transformation process. So let us look into every type briefly. Microbial biomass, for example, yeast production. Yeast is widely used by baking industry, and yeast pellets are produced, and in this case yeast is the product of fermentation. The second is single cell protein. As I already told, between 1960 to 1980, when there was shortage of food, microorganisms were used as single cell protein. So even today they are being produced as single cell protein, mostly for animal feed, and this is one area of industrial microbiology. Next, as such, I told microbial enzymes. Enzymes are the most important product of microorganism which are being used in the industry now. I have highlighted only 4 enzymes. As such, nearly 10 to 15 enzymes are being produced in large scale. The most important 4 types are the amylase, which is being used in baking industry, brewing industry, industry that produces baby feeds, chocolate industry, and textile industries. Pectinase production. Pectinase enzyme as such is used in coffee industry, soft drinks, and industries which are involved in fruit juice production. Protease enzymes has wide application in dairy industry, leather industry, meat industry, and industries which are involved in production of corn syrup and veg soups. Lipase is one more enzyme which has wide application in detergent and laundry industry. Microbial metabolites. Basically, whatever produced by microorganism, we can categorize into 2 types: primary and secondary metabolites. Primary metabolites are those which are produced during log phase. Log phase can also be called as exponential phase, or it can also be called as trophophase. And secondary metabolites are those which are produced during stationary phase, or also called as idiophase of the microorganism. Example of secondary metabolite is antibiotics produced during stationary phase, and example of primary metabolites are amino acids, vitamins, organic acids. All other products come under primary metabolites produced during log phase. So basically, microorganisms produce 2 types of metabolites: primary and secondary. It is categorized based whether they are produced in log phase or stationary phase. Recombinant products, as I told, microorganisms are being genetically engineered now to produce recombinant products. These industries are thriving and they are highly successful as of now. Example, making microorganisms, especially E. coli, to produce insulin. So it is one of the good example for genetically engineered E. coli that produces insulin. Such industries have a huge market now. We call them as foreign products or heterologous products. Examples: interferons, insulin, and human growth hormones. It is one branch of industrial microbiology which is very prosperously growing now. Transformation process, it involves microorganisms ability to follow few biological reactions like dehydrogenation, oxidation, hydroxylation, condensation, isomerization. One classical example is how alcohol is converted to acetic acid in a single reaction. Acetic acid that is vinegar, and it is a single step reaction happening. And there are industries which use alcohol as a substrate and acetic acid will be the product, wherein the microorganism will do transformation process. And one more example is steroid production. Steroids have wide applications, but it cannot be produced in by chemical process because of the complexity of steroid structures. But microorganisms, by using a simple precursor, they can convert them into steroids. So this is about the introduction to industrial microbiology and the scope of industrial microbiology as such. Thank you.